All right, ladies and gentlemen, welcome to another episode of Science Safe Sharks. My name is Rolf. I'm a marine biologist here at Marine Dynamics, close to the southern point of Africa, and I will be your host for today. With me is joining me today is Alina, and she's also a marine biologist here at Marine Dynamics. Hello, Alina.
Hello, Ralph. Hello, everyone who's joining us right now. Thank you so much for tuning in.
Well, yeah, thank you very much. But for those newcomers out there, if you are not sure what we are really up to here, you can visit our Facebook and YouTube sites, see our previous live streams, see what we've been talking about, and yeah, get to know us a little bit more. For today's topic, however, is very interestingly lasers and how to use lasers to study sharks. But before we get into that very interesting topic, so we are a shark cage diving boat, but while we are out there on sea, we collect a lot of data on our animals that we see swimming around our vessels. Now, the very, very important question in this regard, though, is, Alina, why do we collect so much data?
That is a very, very fair question. So as you said before jumping in, how we're going around collecting that data, it's very important for us to understand why we're doing this and why it is so important for all the scientists all over the world to actually do that. So guys, by collecting the measurements, and by measurements, I mean the one that we're going to be focusing today, and that's going to be size, sex, and also sexual maturity. So guys, to collect these measurements, it gives us a better understanding of population of targeted species, therefore better understanding of population structure and their life cycle as well. And this knowledge actually allows us to create and place better management measures and to protect the species. I know it sounds quite easy, those step-by-step way of going around this, but obviously the steps are not as easy as they sound to be. But if there is an exploited species that might be under threat, it is important to actually back up our suspicions with some scientific data.
Absolutely. You can't go around claiming things if you don't have the data to actually support what you're saying.
Exactly.
Now, how would you, what kind of methods would you use to collect the data, the size, the sex, all of that?
So there are different methods to do that, and different methods can be applied to different species of fish. So if we're talking about the aquatic animals, and some species of fish, and when I say fish, I mean our bony fish, some destructive sampling techniques that can be suitable. By destructive sampling technique, I mean fishery-derived sampling and fishery-independent techniques such as scientific surveys. So both of them, they actually require dead individuals. What you need to do, you need to get your hands on the dead individuals and get that measurements and that data collected. But obviously, you are taking some individuals out of the population. So as I said, for some fish species, it can be suitable simply due to the greater fecundity. And guys, for those whose it is a new word for fecundity, it means the ability to produce many, 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 many offsprings. Unfortunately for sharks, it is not the case. You might remember me and Kelly or me and Dickie talking about this, how long it takes for a shark to actually give birth and lay eggs, and how long they live, and actually how long their reproductive cycles are, and that they don't lay or give birth to that many offspring. So it can be quite tricky for the sharks.
Now, what kind of methods would you suggest for sharks in that case?
So with the sharks, to go around collecting the data with them, it would be better to choose non-destructive, non-invasive, and some of them are invasive, so let's just say it non-destructive. And those are the techniques that we are actually using, some of them here in Hansby during our collecting data on the boat when we go out. So we're talking about visual estimates. I actually have a picture how we do that around here. Let me just show it to you. There we go. So guys, you can see that this is a cage and there is a shark just next to it. And we actually do use the cage as a reference. So we do know how long is the cage, and seeing the shark just next to it, it actually makes it a little bit easier for us. However, we both know that we don't really often agree on the size, even when we see the cage, and we do argue about it quite a lot. So there is always an error in this.
Absolutely. It's a very familiar sight that But every time, like sometimes you go like, no, that shark is larger than 3.5 meters, and other people turn around, nope, it's definitely smaller.
Exactly. So there is a definitely bias of the observer, of the person who is collecting that data. The other techniques, there will be the catch and release. And in order to get the sexual maturity, you can go around hormone analysis. But again, as I said, those they might be non-destructive, but they are invasive ways to collect the data. The one that we're going to be focusing on today is probably the most hands-off method, and this is our photogrammetry. So guys, for those, again, if it is a new word, photogrammetry is a process by which detailed measurements can be collected from still image.
All right, so how would that work exactly?
Alrighty, guys. So obviously, you can't get measurement just from a two-dimensional still image. What you need in order to get those measurements, you need a scale bar. And this comes the coolest part of our interview, guys. Here comes lasers. Sharks with lasers. Don't worry, we don't strap the lasers on the shark. What we do, we actually point the sharks, the lasers at a shark to actually create that scale bar. So guys, I'm just going to show you how you go around and how you would set up the method, how to do that. There are the cameras and the lasers. There we go. So you can see that all you need is just two lasers and a camera, and it will work. So it doesn't really matter the quality of a camera as long as you can capture an animal. You can see they are pretty similar looking. There might be different cameras used, different lasers, but technically they are all the same. And yeah, so what would you do? You would point the lasers at a flank of an animal. You need to be at 90 degrees to it, so perpendicular. And you just need to find the flattest side of a shark, and you just point it, and that will be your measuring scale.
So it's very important that these lasers are like perpendicular, or like right parallel to one another, so that you know the distance.
Well, they are two points, so they're going to be parallel anyways. But yes, they just need to be on the flattest side of the shark.
Hi, Tasha.
Hello.
Yeah, laser sharks. So
Laser
I would
shark.
love. Very funny. So what kind of research has this method been used for in the past?
So actually, on the picture that you still see, the bottom right, sorry, almost mixed up left and right, bottom right, this is actually the research that one of our former colleagues, Toby Rogers, he wrote a paper on the laser photogrammetry, and he used it on small spotted cat sharks that are abundant in the UK. So guys, I'm just going to show you the picture of a cat shark, very cute. There you go, guys. So this is how you would go around taking the measurements of a shark. You can see the shark laying very, very nicely in front of us, and you can see the 2 points pointing at a shark, 2 laser points. And this is how it would be in a perfect world. It is a controlled environment, so it is an aquarium. And the GoPro was the one that was taking the footage. So it wasn't taking pictures, it was taking videos, and was just waiting for the sharks to actually get to the right position so they can be directly 90 degrees in front of you. And that's how we do it, go around in a closed environment. And using that, the paper actually found out that measurements we take with the laser photogrammetry is very consistent with the physical measurements that you take from the dead specimen that they collected from fisheries.
All right. So, but this animal is like nicely sitting in an aquarium, and it's a little bit more difficult when it comes to, say, white sharks or other larger shark animals. And especially when you're out in the field, you can't kind of scuttle them into like a little enclosure. How would you go about using this method in the wild?
You are absolutely right. So this is just a little diagram on how you would go around taking what problem you would actually see while taking the image of a live free-ranging animal. So obviously it's not as straightforward as measuring from the tip of the snout to the edge of the caudal, because you actually need to take into the account the swimming behavior of the animal. So you can see that the caudal side, it would move around. So it would actually affect the measurements. So what you do, you would focus on something that stays still, even if it is moving. So with sharks, usually we would focus on their dorsal fin. that you would be just straight in front of us. And using the dorsal fin, we can estimate the total length of an animal. Laser photogrammetry is being used in quite a few researches. On manta rays, also on whale sharks, and even here in Huntsby on our great white sharks as well. There was a laser photogrammetry from surface, so the pictures were taken from the boat.
All right, that's very curious. Now, what does the future hold for such a technique?
Well, it would be useful to investigate whether this technique is suitable for other species of sharks. As we know, all sharks are different, and we know that it can be used for whale sharks and great white sharks and cat sharks, like Toby presented in his paper. However, it is important to estimate the different ratios, body ratios, to actually estimate the length and take the measurements properly. But it can be used for different species of sharks as well. And also the citizen science can be a very good idea with so many divers in the water, especially dive photographers as well. It can actually help and create the huge database that can be very, very useful. And it is, as I said, it would be a citizen science, but still it can create the huge database that can be used in the conservation as well.
Wow, really? So it definitely has some very interesting potential. Would it be useful to use in our area?
I would say so. So you can definitely go around doing research on our sharks using laser photogrammetry. The only problem with that would be visibility, probably. But we do have some nice days. And as I said, there's been research done with our area, with our great white sharks from the surface. So it is possible. It's just the weather and the visibility just needs to be taken into account.
All right, that's very interesting. I'm looking forward to seeing some more about this technique being used. Now, okay, so you've given us quite a nice introduction to this technique. Where would we go if we want to know more about stuff like this?
So guys, the paper of Toby Rogers that I just talked about, it's called the Determination of Size, Sex, and Maturity Stage of Free Swimming Cat Sharks Using Laser Photogrammetry. Guys, you can find it on our Dyer Island Conservation Trust website. So you can just go to www.dict.org.za, and on the About, you can find all our publications, and you can just click on the publications and read them up. And guys, also, if you do want to support us, our work and our research, you're very welcome to do the donations on the website as well. www.dict.org.za.
All right. Thank you so very much, Alina. Yeah, so a lot of the research that we do, we are dependent on public donations. Government, unfortunately, is not very supportive. And nowadays, with the whole COVID pandemic, unfortunately, the tourists are not coming through so much. So, Nikita, welcome back. Nice to see you as well. Yeah, so for those of you, once again, who want to know more about what we do, please follow us on Facebook and YouTube. So if you subscribe, you're going to get notified about new videos that are popping up. So on Friday, and for those of you who do have questions, please ask them. We absolutely love to hear what you want to know and love to answer them. So, and on Friday, we're going to host a Q&A session more about this topic.
Awesome. And also, Ralph, I just wanted to mention as well, for those who are interested in the stuff that we are talking about, and if you have more specific questions or you want to go into a little bit of depth on some specific topics, guys, you're very welcome to drop us a message to academy@marinedynamicstravel.com, and we can arrange the mentor sessions as well if you would like that.
All right, that's great news. And yeah, so another thing that is coming up, that's tomorrow. Marine Dynamics is joining with the local Lamont wine farm, where we're combining certain wines with the Marine Big Five. And tomorrow it's going to be the lovely Lamont Shiraz, and we're going to be chatting about the Cape Fur seal that we get in this area. It is free, but you do have to RSVP. So please visit our Facebook site and you can RSVP there. And then hopefully we'll see you tomorrow evening at 6 o'clock South African time with a nice glass of wine. All
Awesome.
right. Yeah, and that's, I think, it from us. So once again, Alina, thank you so very much for joining us. and also everybody out there.
Thank you, Ralph, so much for having me today. Guys, please ask us the questions, reach out on our Instagram, Facebook, and we are really looking forward to see you on Friday again.
All right, guys, have a very nice evening.
Goodbye. Thank you.